I'm going to tell you about our approaches to try to generate or to understand the pathways that control cardiomyocyte regeneration in the adult heart. This has been a long road. What we use now, our model, is our embryonic stem cells to try to produce progenitors that we think resemble cells in the adult heart, and then look for molecules, small molecules and microRNAs and siRNAs that would drive those cells from one step to the next to produce cardiomyocytes and other cardiovascular cells, ultimately with the idea that we would be able to generate a drug that would stimulate endogenous regeneration. So this is a scheme showing cardiomyocyte differentiation from embryonic stem cells. It's a stepwise process, as everyone knows, and we've been working out many signals over the years that drive each step in this process. So the logic is that there are endogenous cardiac stem cells, and that these stem cells overlap in some ways the cells that arise during fetal development. Not perfectly, of course, but in terms of the profiles of gene expression, there are some similarities. And that has led us to think that if you could have small molecules that would drive one step and then the next step and the next step, you would be able to use these molecules to drive, eventually, endogenous stem cells in the heart to regenerate myocardial tissue. So with that in mind, we've developed assays over the years where we produce progenitors, and those progenitors then are capable at some level, low level, of giving rise to cardiomyocytes or moving the next step. And then we add molecules from one step or another, and then what we read out is a fluorescent signal indicative of cells when they're on track to becoming cardiomyocytes. And you can see here just views from a high throughput microscope screen where you can see that, in this case, GFP indicating cardiomyocytes have appeared in the dishes. And so these are human embryonic stem cell derived progenitors. We make them in bulk at this point, put them in 384 well plates, add compounds for a short period of time, come back a little bit later, and we look for cardiomyocytes. And by flow, if you take one of the hits, and you can see by flow cytometry when you do it in bulk culture, that you can induce cardiomyocytes. So over the last few years, we've done this for different steps in cardiogenesis, and we've begun to populate the differentiation scheme with small molecules that stimulate particular steps in cardiogenesis. And many of these molecules mimic pathways we already knew about, and many of the molecules target pathways we don't know about. And we have many compounds for which we have no idea how they work. And I'll get to that. That's an important point that I'll get to later on. So these are our favorite candidates right now that have come out of the screens. These are molecules we're trying to take forward and develop to the point where they might be tested in vivo to see if they would stimulate endogenous regeneration. So, by the way, this is a movie made by Bruce Conklin of one of our human embryonic stem cell lines for screening, and you can see it differentiating nicely into cardiomyocytes and other cardiovascular cells. And it also, you can see the red fluorescence that we use, that we score in the microscope. So I'm going to tell you about, in the next few slides, a TGF-beta inhibitor that came out of these screens. This is the first selective TGF-beta inhibitor that I'm aware of. And this molecule, I think, has taught us something about cardiomyocyte differentiation that might be applicable to adult regeneration. So I'll only show you a couple of slides on the selectivity of this compound, but TGF-beta-like molecules, there are a number of them, TGF-beta, Int'l, myostatin, and more divergent ones like BMPs and so on. And these molecules all work through a similar set of receptors, a type 1 receptor, ALK4, ALK5, ALK7, in the case of TGF-beta and Int'l, and type 2 receptors, the TGF-beta receptor 2, activin receptor 2, and so on. And this complex signals then through SMAD proteins into the cell. And we noticed with the compound that we'll now call inhibitor of TGF-beta receptor degradation, or ITD1, that it selectively inhibits a response to TGF-beta, but it has no effect on a response to activin, which signals through a slightly different receptor complex, but structurally very similar. And if you look at the level of kinetics here, you can see that there are differences between, we can separate enantiomers of ITD1. We can figure out how well the molecule works on the kinetics. And what I want to draw your attention to here is the Emax, the maximal ability or the efficacy of this compound at inhibiting TGF-beta. And what you can see is that this compound, if you dial it up to, I don't know how far this goes, but 10 micromolar, let's say, you can get nearly complete inhibition of TGF-beta, but even at micromolar concentrations, it does not touch activin very much, only 20%. So it's quite selective, whereas other molecules that you can get from, we don't have them on here, oh, here's one, SB, and they're often listed in catalogs as TGF-beta inhibitors, but they can't discriminate between TGF-beta and closely related receptors. They're kinase inhibitors. So that leads us to the question of how does this molecule work? How can it tell the difference between structurally very related receptors? And when we tested it for kinase inhibitory activity, we found unlike many kinase inhibitors, this is the SB compound here that inhibits very well, our molecule does not inhibit the kinase activity of either the type 1 or the type 2 receptor. Okay? And so unlike all these commercial molecules that block the kinases, our compound in some way was working differently, and what we realized is that it selectively blocked the type 2 TGF-beta receptor. And to make a long story short, what it does is it stimulates the clearance of the type 2 receptor from the surface of the cell. Here is overexpressed type 2 receptor, and you can see at different doses of the different doses of the receptor transfected in, you can see a decrease in the amount of receptor quantified here. Similarly, endogenous receptor in a couple of different cell lines, you can see a similar decrease in the receptor on the surface of the cell. And so how this works, Eric Willems, the postdoc in the lab who did all of this work, devised an interesting assay, a flow cytometric assay, where he looked for the presence of receptor on the surface of the cell and asked how many cells would be depleted of receptor. And so you can, this is an instance here where you see depleted receptor, and you can see in red here cells with the receptor. And so using this assay, he then treated cells with various inhibitors, because we were thinking maybe this receptor or maybe the compound targets the receptor for internalization and degradation. And we thought, well, surface receptors go to the lysosome, chloroquine should block this. But in fact, chloroquine, here's the effect of the molecule itself. Here's no ITD1, you get high levels. You add chloroquine, here you add ITD1, it drops the level down. Chloroquine doesn't do anything. But if we add proteasome inhibitors, MG132 or Bortezimib, you can see that it goes from an inhibited level and it restores now the amount of receptor on the surface. So this indicates that this molecule blocks or targets TGF-beta receptor type 2 for degradation by the proteasome. And that's illustrated here. TGF-beta receptors are known to go to the proteasome, but typically when they go to the proteasome, both the type 1 and the type 2 go to the proteasome. But what I didn't show you is that the type 1 receptors are completely unaffected by this molecule. Only the type 2 receptor goes to the proteasome. We don't know how this works, but this molecule must be stimulating some pathway in the cell targeting this receptor. Okay, so with such a molecule in hand, we then asked, what is the role of TGF-beta as distinct from other TGF-beta type molecules like Int'l in stem cell differentiation into cardiomyocytes? So it's long been known, and as you heard from Gabriella yesterday, that Int'l is very important for producing cardiac progenitor cells. This is just fundamentally important for producing all sorts of mesoderm in the body. But once you get to this progenitor, what happens then? How does the progenitor go to become cardiomyocytes, smooth muscle cells, or vascular endothelial cells? And what I didn't tell you is that Int'l initiates a cascade. So Int'l activates itself, but Int'l also activates TGF-beta. And then there are feedback inhibitors that suppress Int'l, allowing TGF-beta to persist in the embryo or in the stem cell cultures. So at about this time here, during development, there's very little Int'l, but there's a lot of TGF-beta. So by adding the small molecule at this window of differentiation of mouse or human embryonic stem cells, what we noticed is the small molecule was able to increase cardiogenesis. And just looking at the human data right here, you can see this is the fold increase. You can see about a 75, 80-fold increase in the number of cardiomyocytes in the cultures when you add this small molecule at this time window here. And you can see a slight but measurable decrease in endothelial cell markers and smooth muscle cell markers in the same culture. That's more pronounced in the human than it is in the mouse, by the way. So what we conclude from this is that TGF-beta suppresses this branch of the cardiopoietic differentiation pathways, and that TGF-beta, although not diagrammed here, promotes these branches here. So thinking back to Ken Chen's talk yesterday, when he was showing that VEGF would induce the formation of multipotent progenitor cells or enhance their formation, even in vivo, and he was asking what might be acting subsequently to apportion cell fate from a cardiopoietic progenitor. I would argue that it's signals like TGF-beta that play that role. So in trying to understand how this works, as a graduate student in the lab, Wenqing asked the question, what is the critical switch that commits a cell to becoming a cardiomyocyte? So we were strongly influenced by work from a number of laboratories, including our neighbor Lorenzo Puri. And he and these laboratories have been looking at a complex called SWI-SNF, which is a chromatin remodeling complex. SWI-SNF itself has no transcription factor function, so the complex is taken to the promoter through interactions with transcription factors where it opens the chromatin, allowing transcription to ensue. So, and as you're probably mostly aware, Benoit Bruneau took advantage of this idea to ask whether BAF60C and cardiogenic transcription factors like GATA4 and TBOX5 could reprogram cells in the embryo or in stem cell cultures to becoming cardiomyocytes. And this is just one figure from his paper where he showed ectopic cardiogenesis, and that's indicated by these, here you can see the ectopic formation of cardiac cells. I forget the marker that was used here. Oh, NKX2.5. You can see ectopic NKX2.5 in response to injecting constructs that would express the chromatin, the key subunit of the chromatin complex, BAF60C, along with these cardiac transcription factors. And this then brings that whole complex to activate cardiac gene expression. Okay? So we then looked during stem cell differentiation to ask about these particular BAF60 variants, A, B, and C. C being the one that's most muscle prone. And so what we noticed is that during differentiation that there is an inverse relationship between BAF60C and A and B, so that all A and B are more prominent early, and BAF60C comes up very briefly, a spike of BAF60C expression. And this spike here coincides with the time that multipotent progenitors become committed to the cardiomyocyte lineage, leading to this model that perhaps this complex is what says B cardiomyocyte. And we noticed also that we wanted to know what would regulate this, and we noticed that this occurs just as the natural TGF-beta nodal BMP inhibitor, Cerberus, is declining in these cultures. And we had years ago discovered that Cerberus, this came out of a small protein screen, so Cerberus is a very strong inducer of cardiogenesis. And here was some embryological data from Xenopus embryos showing that if you overexpress Cerberus in a region of the embryo here in red, that you could induce cardiogenesis at a distance away. So Cerberus doesn't directly say B cardiomyocyte, it does something that says B cardiomyocyte. Okay? So Wenqing then found that Cerberus, if you knock down Cerberus with an siRNA, you can come back and rescue it by BAF60C overexpression. Furthermore, if you knock down BAF60C, you can see a reduction in cardiogenesis. And then finally, if you knock down Cerberus, of course, it also blocks cardiogenesis. So this gives you the idea that Cerberus might be inducing cardiogenesis through the action of inducing BAF60C. So since Cerberus blocks nodal and BMP, Wenqing then went on to ask, how are these related in a signaling cascade? And what she showed here, if you treat cells with activin or BMP, activin is a mimic of nodal. If you treat cells with activin or BMP, or inhibitors of that process, not the chemical I told you about, ITD, but the commercial version that blocks both nodal and TGF-beta, or a BMP inhibitor, well, you can find, if you look at these graphs, that nodal TGF-beta block BAF60 and GATA4, and BMP blocks GATA4 and TBOX5, giving you the beginning of a cardiogenic signaling hierarchy. Okay, and you can see that even more dramatically when you look at Flick progenitor cells here, and you can see that the siRNA to Cerberus will give you a reduction in BAF60, GATA4, and TBOX5. And that leads then to this overall cascade that Cerberus, through the control of the nodal TGF-beta cascade and BMP in cultures, controls a set of cardiogenic transcription factors and the chromatin complex. Okay, to pull this all together, is this true? Does Cerberus actually remodel chromatin at cardiogenic genes? And so we took advantage of the fact that the NKX 2.5 promoter has the early part of the promoter, the part that's responsible for turning on NKX 2.5 within these early cardiac progenitors. It depends upon an element located at -9 kilobases from the start site of transcription. This has been well known. And nearby, there's an element at about -3 kilobases that's important for endodermal organs. So we asked whether, if you block Cerberus, would the chromatin around those two elements become closed? And we used a chromatin accessibility assay in which you take nuclei and you digest with an enzyme, and the accessibility of the enzyme to chromatin gives you a readout of whether that chromatin is open or closed. So the result of that is shown here. Here's the diagram again of the promoter. And here we looked at the -9 kb region and the -2.8 kb region. So if you look at undifferentiated ES cells in black, here, this is the chromatin for cardiogenic genes is off, it's closed. If you look at, in the gray here, at differentiating cardiac progenitors, you get this amount of accessibility. You get less DNA recovered, so the chromatin is open. If you block, then, with BAF60C RNA or Cerberus siRNA, what you see is the chromatin goes from being open to being closed. Okay? But if you block with GATA4 or TBOX5 alone, either of them, it has only a modest effect. So basically what this is saying is that BAF60C and Cerberus are critical for opening the chromatin. Furthermore, if you have a block of Cerberus and you add various molecules back, can you open the chromatin even when you've blocked Cerberus? So here are the two controls as before. Here's the closed chromatin in red here. It closes when you have Si to Cerberus. And then if you add BAF60C, it opens again. BAF60 plus GATA4 also open. BAF60 plus TBOX5 open. And here, interestingly, when you leave out BAF60 and you just add the transcription factors, it's somewhat intermediate between the two. And so we don't fully understand this. We think there's a network between the transcription factors and the BAF complexes so that you get some BAF induced, and the knockdown is only partial. So I really don't understand this last result completely yet. But what this does tell us is that these three molecules seem to work together downstream of Cerberus to induce cardiac differentiation. Okay, so that's what I've told you here is the model, that other Cerberus and in the embryos, other inhibitors of TGF-beta and BMP, like Noggin and Lefty, would regulate these signals, inducing this transcription factor and chromatin complex to then open chromatin, and that's how you would commit a cell to becoming a cardiomyocyte. Extrapolating then, this is again what I've told you, our molecule, which blocks TGF-beta, would work at this level, perhaps by doing the same thing. And if you were to go to the adult heart and translate this then to the adult situation, you can imagine that it might also take some progenitors and commit them to the cardiac lineage by blocking TGF-beta. And as you know, the scar formation, or the region of scarring and fibrosis, is thought to be hostile for stem cell differentiation into cardiomyocytes. So I'm proposing that if you were to dial down TGF-beta, perhaps as well other molecules involved in fibrosis, say PDGF, what you would be able to do then would be to create a more an environment that is more hospitable to creating cardiomyocytes. So we're testing that in vivo now. Okay. And that's basically what I've told you here. So I'd like to make the transition now and talk about more complex screens. This is a very simple, high-throughput pharma-style assay that we used, where we look for small drug-like molecules. And I think actually that after having done this for a number of years, that's a big mistake. Because once you get a small molecule out of these screens, it's very difficult to figure out the target. We don't even know how ITD1 works. We don't know the direct target of the molecule, and we've been working on it for years. So you do not get a systems-level understanding of a complex biological process by studying drug-like molecules. So I think it's better to screen molecules like microRNAs or siRNAs or focused libraries that target pathways to get pathway level understanding of the problem. And then you can figure out what you want to target as a drug. So that also works well for more complicated assays. Typical pharma assays are very high throughput, but then there are more complicated assays, such as, this was actually Stefan Engelhardt's assay for hypertrophy here, where you can screen, it's basically a low throughput or moderate throughput assay, just like stem cell differentiation. And you can even think about assays for physiology that I'll tell you about in a second. So, as a proof of principle, we went back to the embryo, and Alex Colla, a postdoc in the lab, made, again, mesodermal progenitors, and asked, can we use microRNAs to dissect the pathways that initiate cardiogenesis? And the initiating step in cardiogenesis is to make a cardiac cell versus a gut endodermal cell. That's the first step to make a cardiomyocyte. And so he built an assay that could distinguish between these two cell types, and he screened the whole genome's worth of microRNAs. And that's just outlined here. So you do the microRNA screen, you basically get screen results. The best hits are shown here. He used an informatics approach to assemble what he calls, or others have called in the lab, a reticulome. You get this god-awful network. And then you try to make sense of that by screening for siRNAs that block all the interesting targets in here. And by doing that approach, he ended up with TGF-beta signaling, not surprisingly, as the molecule that as the signaling pathway that can regulate the switch between endoderm and mesoderm early on. So this for me, I was thrilled with this. This was great. It shows that screening microRNAs, you can go from the screen result to getting pathway information. I'm going to digress for just a couple slides to show you that that two of the microRNAs that he discovered are actually normally involved in governing the difference between your gut and your heart. And so he went to mouse and frog embryos. I'm only showing the frog data here because it's easier to understand. But the mouse data is very similar. So that normally, this part here that you can see stained on the uninjected side of the embryo is the endoderm here. And if you inject the let's see what this was injecting. Oh yes, the microRNA was LET7, and he built a target protection oligo. And so here's the LET7 bound to its target, which is the TGF-beta receptor, or the activin receptor, the nodal receptor. You put in a target protection oligo that masks it. Now the microRNA cannot interact. If you inject this oligo onto one side of the embryo and a control oligo onto the other, you can see that the cells that are normally mesoderm, over here, the clear area, now become endoderm. And that's seen also with looking at another endodermal marker, and the mesodermal marker retreats and goes away. So this blocking just the interaction with between LET7 and the target will then switch the fate of these cells. And he's done this with many other markers. Okay, so this is the idea that presumptive embryonic cells that don't know whether they're going to be mesoderm or endoderm are patterned by the expression of LET7 and miR-18. And those then block the responsiveness to nodal, giving you the difference between mesoderm and endoderm. Okay, so, but what I really want to focus on is that if you have a complex biology, say a disease in dish assay or stem cell differentiation, you can screen against various focused libraries and get a systems level understanding of the process, and then take those targets into traditional pharmaceutical company type drug screens, hopefully. Okay. So I'd like to conclude with a few slides now just on looking at a very slow form of screening, and this is to look at cardiac physiology. I've been very interested in the problem of what stimulates the maturation of cardiomyocytes, and also what controls the contractility of cardiomyocytes. And of course, much is known about contractility, very little is known about maturation. And so what we've been thinking about doing over the last few years has been to develop assays where we can score the physiological function of stem cell-derived cardiomyocytes in order to understand pathways that control maturation and contractility. So it's this last part here in the differentiation program. So with engineers, in particular the laboratory of Jeff Price, who founded a company, Vala, now to commercialize the instrument, we've developed an instrument that can automatically look at the physiology of the cells. It makes movies of this field. Here we're showing calcium transients. And it basically is a high throughput microscope that records this image and then segments that image into images of movies of individual cells, and you can get kinetic data. So it has a lot of advantages here that I don't need to labor you with. This is now the first commercial version of this instrument that's just being produced now. And it has a routine where it images the cells, captures those images over time, and then the best part of the instrument is software that segregates the image into images of individual cells so that you can get kinetic data, calcium transient data shown here, for every single cell in the field of view in just a matter of seconds. Okay? In the time it takes basically just for the calcium transient to occur. And you can get, of course, all sorts of kinetic data. So why single cells? Why bother doing it with single cells? And that's illustrated here. I showed you before a beautiful movie of synchronously beating cardiomyocytes. But if you put in a compound that's arrhythmogenic, say sparfloxacin here, you can, depending on the compound, see asynchrony develop in the culture like this. And you can imagine that these 3 different cells, for instance, are beating at different times. So, and that's shown, and you can see that that's shown here in the calcium traces. So that if you were to take a whole well view, okay, you would in essence just get a flat line because of all the individual cells basically are canceling each other out. So you get kinetic data looking at every single cell, plus you can look at if you had a culture where there was pacing activity, you can get conduction velocity looking from one cell to the other. So you can get a lot of interesting kinetic parameters by looking at single cells. So looking at the effect of a few drugs, we've looked at a number of drugs to try to validate this with the idea that this would also be useful for arrhythmogenicity testing of drugs early on in the pharma pipeline. But here we can look at a channel, a calcium channel opener or closer. Here, verapamil to close the calcium. You can see that blocked. The opener here, Bay-K. You can see how it increases the height of the transient, but most importantly, the timing of it. If we look at molecules that have effects on QT interval, you can see similar effects, interesting effects to fetolide, which directly blocks ERG. You can see effects on the calcium transient. And in particular, you can see here a little blip indicative of after depolarization, we think. Here, flecainide, which also has, besides sodium, has an effect on ERG. You can see its similar effect. And this is a nice, we have nice dose responses for the calcium transient effect of these various drugs, as you can see here. We can do it in either a paced mode, where the electrodes automatically, the robot puts electrodes into each well and paces the cells, or we can look at spontaneous beating. So we've been comparing this instrument to looking at the ability of this instrument to discern changes in the calcium transients of a number of drugs that are known to, either safe drugs or drugs known to be arrhythmogenic, and looking to see whether the IC50, EC50 values that are reported compared to the EC50 values that we can detect. And what you can see in this table is that they pretty much line up. We can match or in some cases beat the reported IC50s for a variety of drugs. Okay, so we're now moving on to be able to use this instrument to record voltage simultaneously with calcium by putting a second camera on this instrument. You can see here 2 cameras in the light path. And here are our first voltage recordings. And you can see here that when we superimpose the two, you can see the calcium transient and the voltage transient. And before everybody gets up and tells me that they should not be superimposable in the upstroke, I remind you of 2 things. Firstly, these are immature cells. These are these are human iPS-derived cardiomyocytes, which might have calcium-dependent upstrokes, firstly. And then secondly, the camera that we used on this recording is only 33 hertz, so might not be able to distinguish the fast sodium channel upstroke from the calcium upstroke. So we now have a 100 hertz camera on this instrument. Okay, so applications of this system, and this is the last data slide. I'm very interested in this problem of maturation of cardiomyocytes. So if you look at a profile of one parameter of the calcium transient, I believe this might be the upstroke speed or the width of the calcium transient, I can't recall. But you can get multiple parameters. We can get the Vmax, we can get times to decay, we can get the decay max, we can get the amplitude, the full width half maximum of the transient. So if we look here, I believe, as I said, this is the full width half maximum or the upstroke velocity. What you can see in the dark colors here is what happens is the profile of cells, whether they're in the first 5th percentile, the last 10th percentile, and different percentiles. You can see the distribution of cells and how fast they are. And so untreated cells here give a nice profile, okay? Basically a log normal distribution. And if you look at endothelial conditioned medium treated cardiomyocytes, these are human iPS-derived cardiomyocytes treated with a conditioned medium from either HUVEC cells or a microvascular endothelial cell line for about a week, what you can see is that drives the distribution of cells so that they have faster calcium kinetics. Now, if you look at TTX sensitivity, okay, so have they acquired sensitivity to a sodium channel inhibitor? So in essence, is the upstroke now becoming dependent upon TTX-sensitive channels? What you can see is that early on, iPS-derived cardiomyocytes, as they come out of the cultures, these are only about 2 weeks old or 3 weeks old, there's no TTX sensitivity that's apparent. However, there's dramatic TTX sensitivity that's apparent if you treat these cells with conditioned medium from endothelial cells. So I don't know the factor yet. I believe it's less than 3,000 molecular weight based on preliminary data. But what I'm using this for is an illustration that we can use this instrument as an assay to identify molecules that stimulate the maturation of cardiomyocytes, which has long been my goal. Okay. So in summary then of kinetic imaging cytometry, I think we're now successful at having an instrument that can read out high throughput physiology. And then, of course, this will have many applications, especially in the disease in dish types of assays that we heard about yesterday. So I'd like to acknowledge the people who have contributed to this. In particular, a couple of postdocs, Eric Willems, who did all of the Eric Willems, as well as a graduate student, Paul Bushway, did all of the small molecule screening work that I told you about today. Eric is actually on the job market if anyone needs someone who is good at this sort of thing. He's keen on, he might want to return to Europe. He's from Belgium originally. And also Alex Colas, a French postdoc in the lab who did all of the microRNA work, and Wenxing Kai, a graduate student who did the BAF60 work. As well as, of course, this couldn't have been done without chemist colleagues. We make hundreds of analogs of these molecules, make enantiomeric, separate the different enantiomers when they're stereo enantiomers. And this is a huge amount of work done by my chemistry colleagues, and of course the engineering colleague, Jeff Price. Thanks.